forensics, 28 forensics and 24 quality control. So make sure you guys tune in for those as well. So now we move on to the topic for today. As I said, I'm Dr. Payush Dayal, working as a research scientist at HiMedia in proteomics department. And the title for today's discussion is Protein Purification, its concepts, techniques, and applications associated with it. So you guys must have read or heard or studied for DNA, RNA extensively, especially within the COVID years, mRNA vaccines, how do we extract them and everything. But what I'm here to tell you is the future of biochemistry, of biological sciences lies with proteins. So that's why this lecture is important for the next 20 years when it comes to a protein and what the implications and what it would be useful for. So why do we actually purify proteins? We purify proteins for various reasons: to study their structure and their functions, of course, to investigate the enzymatic activity and understand the disease-related mutations that may happen. And in biotech and in biopharma industry, the purified proteins are used as medicines, of course, like insulin or monoclonal antibodies. They're also crucial in diagnostics as well, for example, for ELISA kits and for COVID test kits as well. And the last one is for general industrial usage, like as enzymes in your everyday use detergents and in your cheese making processes as well. And one more part is your biofuels, which would eventually what people in our field believe would be the future as well. Like biofuels are hopefully the future. We'll see how it works out. So now we move on to basic workflow of protein purification, a standard purification like portfolio or a workflow. So we begin with sample preparation, where cells are grown and harvested, of course. If you are a BSc student, you might be starting to understand how do we grow cells, even before that, like how do we express proteins inside of a plasmid and such. But over here, we are straight away jumping towards, we already have expressed protein and we are growing inside of cell. And now we will purify it from that cell part to our actual wall part. So that's where the sample preparation part is, where cells are grown and harvested. Then we do cell lysis, where we break open those cells in order to release the proteins. And then we have a crude separation. It's called like a cell lysate, where you got everything in there. You got your target protein, your protein of interest, but you got whole other things as well, and they're a nucleic acid contamination as well. So that's your crude part. And then we move on to your purification step, because once something is crude, you have to purify it, because you have to go to your target protein part. So then we'll cover the purification steps per se, by charge, by size, by binding specificity, which is affinity chromatography and such. And next we will concentrate that protein, because you have to make sure that protein is at a particular concentration for a particular purpose that you're going to use it to. And buffer exchange part as well, you will learn hopefully. And then you'll do QC, the quality check of your protein, that is it actually the protein that you actually need or not. So now key protein properties that affect purification. Like protein is an entity in itself. It has its own properties, like any human. Human has its own entities. Protein itself has its own entities as well. Protein has a size, of course. You have a weight as a human. Protein has a weight as well. Bigger proteins move more slowly towards gel and columns, as we will see during the course of this presentation by using SEC. The next part is charge. Charge is associated with proteins as well, whether they're positively charged, or negatively charged, or what have you. And then will the solubility of the protein as well, affected by the salt that you use in your buffer, the pH of your buffer, the temperature associated as well, like in your surrounding. And proteins may precipitate or aggregate. They might not be soluble at all. Or, then we can use different techniques like salting in or salting out. And then the binding affinity in order to purify that protein. So over here, like affinity chromatography, we could try, and hydrophobicity is another one. Then other are like IEC and such as well. So these are the key properties of any protein you need to first understand before you start to purify them. What's the size of a protein? What the charge associated with the protein is as compared to its pi, which is the isoelectric point, and the solubility of a protein, its binding affinity, like if actually has a tag associated with it or not, and how many hydrophobic amino acids are associated with it as well. Can you actually use HIC, which is like hydrophobic column, or not? So now let's go to first thing that you do with a protein, salt precipitation. Now, salt precipitation is one of the oldest and most economical method as well. That's why it's widely used. This technique relies on the principle that protein solubility decreases as salt concentration increases. As we slowly add a salt, like in this case, ammonium sulfate, the water molecules that normally solvate the protein get sequestered by the salt ions. Now, this forces the protein out of solution, causing them to aggregate and precipitate out. Now, by carefully adjusting the saturation levels, we can selectively precipitate a protein target based on their solubility. Now, some proteins come out at 40%, some at 70%. Usually it's like 30 to 70 as well in some protein cases as well. Now, once you have precipitated and collected your protein, we often need to remove the excess salt associated with it. That's where dialysis comes in. Dialysis uses a semi-permeable membrane to retain the protein while allowing the smaller molecules like ammonium sulfate in this case to diffuse out the membrane. Now, this not only dissolves the solution but also exchanges the buffer as well to your desired buffer, preparing the protein for more sensitive purification steps like we will see in future, like affinity, ion exchange, hydrophobic column, what have you. So in summary, this step is a combination of like salt precipitation for bulk purification and dialysis for cleanup and buffer preparation as well. Next thing you could use to purify protein is size exclusion chromatography, or SEC. It separates proteins based on their size and shape, of course, hydrodynamics shape, just to be clear, using a porous resin. Now, large proteins are excluded from those pores and they travel faster through the column, eluting earlier. Smaller proteins enter the pores and they take a longer path, hereby eluting later. Now, this technique is gentle and doesn't require harsh chemicals because you could usually do it with just a trace buffer. So it's ideal for your protein of interest, and it's commonly used to remove aggregates and multimers or degraded fragments. So why do we use SEC, size exclusion chromatography, to remove first and foremost aggregates from your proteins? Like consider if a protein is a monomer, for example, but it's aggregating into a tetramer or a trimer. You want to remove that out because you don't want that. Or it's just like aggregating, like some part of it is aggregating as inclusion bodies. So you want to remove that out as well. So that's why we use this technique, SCC. So SCC follows affinity purification to help isolate the corrected fold protein, whether it's dimeric form or form of your choice. Now some of the common SCC columns that you may have used would be Superdex 200, Supro 6, or Cefacryl S100, depending on your molecular weight of your associated protein. And the next is ion exchange chromatography. Ion exchange chromatography separates protein based on their net surface charge. Proteins bind to resins with the opposite charge, either positively or negatively charged, depending on the pH of the buffer and the protein's PI, isoelectric point. Now, for example, at a pH below its PI, a protein is positively charged and binds to a cation exchanger. Elution is achieved by increasing the salt concentration or adjusting the pH, which disrupts these ionic interactions. Now, IEC is widely used due to its high resolution, of course. Resolution is something that associated with your column. When you look at the peaks, like resolution of your peak. Or scalability. Scalability is like if you want to increase your like protein concentration or your protein volume, and ability to separate closely related isoforms or charged variants, like negative variants, positive variants, what have you. Now, ion exchange chromatography is of two different types. Cation exchange, which uses negatively charged resins like Nuvia S, I think it's one of the most commonly used column, which binds positively charged proteins when the pH is below the PI. Now these proteins are eluted by increasing the salt or raising the pH, and then there is anion exchange, which uses positively charged resins such as DEAE, which is I think one of the most commonly used columns as well. A lot of you, especially the MSc students, would have heard about it, which binds proteins that are negatively charged at pH values above their PI. Elution is done by lowering the pH or by increasing the salt concentration, as you've seen with cation action as well. Now over here we have a little bit of a product placement. So we do have our own kit, a teaching kit, a hyper ion exchange chromatography, which demonstrates the purification of lysozyme from chicken egg white using ion exchange chromatography and subsequent estimation of the enzyme activity. So if we have any people in audience who actually like teach classes or something, then they could use this kit to teach them how the ion exchange chromatography actually works. Then we have affinity chromatography. Affinity chromatography is a highly selective technique that relies on specific interaction between a protein and a ligand attached to a resin of your chromatography column. For example, a histag protein binds to nickel ion or nickel NT column. Over here you could see that nickel NT column. This method is useful because it can purify protein directly from a crude lysate with minimal steps, because the nickel NT column is directly going to target your histag that's associated with the protein, and just going to form that bond with it straight away. Now, this is usually, if you have a protein that requires multi-step purification, affinity chromatography is usually the first step that you do, because it usually washes away a lot of non-specific proteins, or not of the proteins that are done in like background expression as well, and it actually just like targets your own protein of interest, because usually you do know your own protein has that his tag when you expressed it. So, over here, so then when you just like crude lysate over here, you do know that only your own protein is binding to that column. So after washing step, unbound proteins go away. The target is eluted, usually using a competitive ligand like imidazole. Over here, usually we use imidazole concentration between 200 to 500 millimolar, depending on your protein, of course. Or there are different ways to use it by changing pH as well, or salt concentration as well. But imidazole, changing for nickel NTA, changing the concentration of imidazole actually works from like your equilibration step to your washing step to your elution step. So what the standard protocol is, your equilibration would have 10 millimolar imidazole inside of it, so as to avoid any non-specific interaction with the column. Then wash step would have 30 millimolar imidazole, so as to remove again any non-specific stuff that's binding very loosely to your nickel NT column. Then we move on to 250 or 500 millimolar imidazole concentration, where the imidazole would actually replace those bonds that have been binding to your protein, and your protein would get eluted out. And then we have hydrophobic interaction chromatography. It separates proteins based on their surface hydrophobicity. Under high salt conditions, water molecules are excluded, and hydrophobic regions on protein interact with hydrophobic ligands on the resin. Now, as the salt concentration is gradually reduced, proteins elute based on how they bind. Now, this method is especially useful for membrane proteins. That explains between the your membrane region of your cell line of your cell, or protein with exposed hydrophobic patches. Now, common HIC resins include phenyl, butyl, or octyl cephros, and the technique is often used as intermediate purification step. Now, this is usually this is a kit that we use for like monitoring the purity of a protein. So HiMedia actually provides this is again a product placement for SDS page kit that we do provide, the equipment, the apparatus that we provide. Now, SDS page is a fundamental technique to assess protein purity. Of course, it separates protein based on molecular weight under denaturing conditions, like for example using SDS, which gives protein a uniform negative charge. Smaller proteins migrate faster through the gel. A single sharp band usually suggests that high purity is achieved over here, in this case like for 28 kilodalton, as you can see, while multiple bands indicate contaminants or degradation products. As I said in this example, we had like 28 kilodalton seen consistently in our elution, shows that the purification step actually worked. Now, continuing with the monitoring your purity with the SDS Western blot as well. Now, Western blotting builds on SDS page, of course, by transferring those proteins onto a membrane and detecting the target using specific antibodies. So once you run your gel, you want to transfer those particular proteins onto a gel as well. So you run it through a current, and you have different antibodies associated with your particular protein. Like for example, if your protein has an antibody against his tag or a MIG tag or a flag tag per se. So over here, like what we usually use is like just like his tag. So we usually use the anti-his antibodies, and it's extremely useful when multiple proteins are present, because then you could actually just like classify, because we do know that our protein is the only one that has anti-his tag attached to it. So has a his tag attached to it, so it's only going to bind to the anti-his tag antibody. A strong signal at the correct molecular weight validates your successful purification. Now this is especially important working with tag proteins, for example, for sure, like his tag, GST tag, MIG tag, flag tag, what have you, or with biotherapeutics proteins. Okay. So now, another step for monitoring the purity of purified protein. Now, protein concentration is often estimated by measuring absorbance at 280, since aromatic amino acids like tryptophan or tyrosine absorb at UV light. A pure protein will typically have a 260 by 280 ratio less than 0.6. Why is that? Because that's when you know your nucleic acid, your DNA, RNA, your background stuff in there, it's lower. Your protein is pure. So that's why we focus on, whenever you are purifying protein, not I focus on, but like everyone focuses on, just to make sure that your absorbance 260 by 280 ratio is less than 0.6, indicating minimal nucleic acid contamination. Now, if the ratio is higher, you suggest RNA or your DNA is still present. So your purification didn't work so far. Now, this is quick and a non-destructive method for your routine quantification of your protein during your purification step. Now, you have to do your activity assay as well. Okay, you have purified a protein. How do you know it's your protein? Okay, you have done Western blot, you have done SDS page, you have done absorbance study. You do know from your from STC, from your IEX, what have you. Okay, your protein percent. But if your protein is an enzyme, actual enzyme that actually performs a process in there, it actually binds to a substrate. How do you know? You do activity assay for it. So activity assays are crucial for confirming that a purified protein is functionally active, because especially if you are purifying an enzyme from monooxygenase family, dioxygenase family, from CDO, what have you, your protein is only valid if your protein is active, if your enzyme is actually active, not just present. Anything could be present. In its denatured form, it could be present. In its inactive form, it could be present. But for enzymes, this means checking substrate conversion or your product formation as well. So over here, we have an example for alkaline phosphatase, hydrolyze, alkaline phosphatase from E. coli, which hydrolyzes PNPP to P-nitrophenol, which shows a yellow coloration once the reaction is complete at absorbance 405 nanometers. Now, increased absorbance directly correlates that our enzyme is active. Now, these assays help evaluate that our protein is correctly folded, first and foremost, the integrity of a protein is there, and effectiveness of a protein. Post-purification is there as well. Now, continuing with the monitoring of a protein, you do, okay, so far I've talked about the protein purification techniques, how do you purify it based on different multi-step processes, and how do you know if your protein is worthwhile by using SDS page or by using the activity study or Bradford assay if it's still there. But how about we actually try to purify a protein? We do a case study. So over here, we do purification of recombinant glucose 6-phosphate dehydrogenase, G6PD purification. It's a key enzyme in the pentose phosphate pathway. The target is human GCPD expressed in E. coli in our case, using a PET vector, PET vector, with his tag attached to it, of course. And it's in a subunit, monomer, weighs around 59 kilodaltons. And the enzyme functions as a dimer, though. Like monomer weighs around 59 kilodalton, but it exists as a dimer. The goal is to obtain a highly pure and active form of a protein. Now we'll use a multi-step purification process, starting with, of course, ammonium sulfate precipitation, which we talked about, followed by nickel NTA, affinity chronography, then size exclusion, and finally ion exchange. At each stage, we monitor the progress using Bradford assay, of course, if your protein is still there, SDS page, of course, and enzyme activity assays as well. Now, over here, so target protein is J6PD, molecular weight 59 kDa. Expression system is E. coli, BL21D3 with PET vector, as I already talked about. Application is important. Like, why are we actually purifying this protein? Is the protein actually worth it? There are like millions of proteins that we could purify. Why this? It's in pentose phosphate pathway. BSc student must have heard about it. MSc student certainly know what pentose phosphate pathway is, and it is medically relevant as well. Its deficiency is not good for human system. So now we move on to our objective: to purify an active recombinant protein. The steps that we would use are, as we discussed before, ammonium sulfate precipitation. Second would be affinity chronography using nickel NTA in this case. Size exclusion chromatography would be next. IX would be the last one, and then we monitor our protein. That will be a QC step to know that we actually do have protein or not. Okay, so in the first step, we use ammonium sulfate precipitation to concentrate the protein and remove any loosely bound impurities. Proteins precipitate at different concentration, so we slowly add protein, and in case of J6PD, it's a very well purified protein. So we already know at around 60 to 70% is actually precipitate side, while keeping it solution cold. So we So at 70%, we do know that it precipitates out. Okay, so we are concentrating protein first and foremost at 70%, so as to make sure that all the other things that goes away from protein. We lyse our cells, bacterial cells, and ammonium sulfate. So I think we got confused over here. Okay, and then once we have that solution, bit concentrated, we do know that we have actually taken out a lot of things that we don't need. At salt precipitation, we do affinity chromatography, because when we make a plasmid, when we make a clone, we make sure that we have a his tag attached to them as well. So then we do affinity chromatography. In this case, nickel NTA. So over here, you could see that the pooled LOE after your nickel NTA shows that we are actually purifying this protein. Now, when you load on nickel NT column, there's going to be a flow through, of course, with any column associated with it. So your equilibration buffer over here, I usually tend to have some imidazole in it as well, so that non-specific interaction is going to be tackled in beforehand. And then we do a wash buffer with 30 millimolar imidazole, so that some non-specific interactions that were there could be tackled with a wash buffer. And then you do elution. That's when we actually know that we are actually dissolving any our own protein that's in there. So procedure is like load resuspented protein on nickel NT column, wash with low imidazole, and elute with gradient. Now, once you've done that, you move on to size dilution chromatography. Over here, we have used Superdex 200 because of the molecular weight of a protein. Superdex is actually pretty good for 100 to 600 kilodaltons. So that's why we have used Superdex in here, because our protein is actually at 118 kilodaltons. So we do know that our protein falls behind that, between that range. Now, your principle is based on, of course, separation based on size and shape, as we've discussed already, and the concentrated eluted fraction would then be loaded onto affinity chromatography. Now, SCC, before we move on, SCC, as we discussed before, and we are going to discuss again, for this protein, it actually preserves the protein structure because no harsh buffers are used, making it ideal, and it's pretty gentle as well. It just separates out based on size as well. So once we have that, the eluents from this part are going to move on to your ion exchange chromatography. Now, in your ion exchange, over here we're using an ion exchange chromatography with Q-Cephros column. Now, why do we use Q-Cephros column for G6PD? Because it has a pI of around 6. So at pH 8, it carries a negative charge and binds to the positively charged resins. Now, we start with a low salt buffer and apply a gradient of NaCl to elute this protein out based on the charge strength. Now, this step provides high resolution separation of isoforms, degradation products, or proteins with similar size but different charges as well. So now over here, why this particular purification order, which got a bit confused in the middle, but let's just address that. The purification order actually matters depending on every single protein. Over here, we started with salt precipitation, because it's, first and foremost, is inexpensive. It's not that hard to do, and it removes large amount of your impurities. The next, we move on to affinity chromatography. Gives the strong enrichment based on the his tag, and then SCC is used because affinity has a low affinity has a binding capacity, but it doesn't actually segregate based on size. and aggregates. So then we could use SCC for that. And then ion exchange offers charge based. So for our case, we use ion exchange for polishing, giving it a very refined product. So that's why we used these particular steps. Now, then for a purity and activity assessment. Now, throughout your purification, we use like when I was a PhD student back then, I would do Bradford assay after every step, just to see whether we actually are getting protein of interest or not, enzyme interest or not, specific activity or interest or not. So, like we use Bradford assay after every step, but over here, for total concentration and to calculate specific activity associated with our enzyme. Now, for functional validation, the G6PD activity assay measures NADPH formation by monitoring absorbance at 340 nanometers. The more NADPH produces, the more active your enzyme is. Now, tracking specific activity across steps tells us just how much protein we have, because you can't have an end product without an enzyme, without your protein, and like how pure or how functional it is as well. This is our like final result for the case study that we did. At the end of purification, we could say that when we had like a crude lysate where protein was like 100 milligrams, for example, but your specific activity was low because your actual protein of interest was in low concentration. There were a whole bunch of other things in there in a crude lysate. Then they did salt precipitation study. Your total protein got low, but your specific activity got higher. And your SDS page as well, you got fewer bands. Then you did affinity, further purification, went down low. Again, total protein at 20. You're going towards your own protein of interest, and your specific activity keeps getting higher as well. And now you're starting to see a clear band at 59 kilodaltons, which is your protein of interest. Then it is SCC, size occlusion, and your total protein went down 15, but your specific activity keeps on rising, and your bands start to get clear again. Then IEX, which is for us was polishing step, like last step that we do. But in some cases, SCC is a polishing step, not IEX. But in our case over here for G6PD, IEX was more. And then over there, you see that specific activity reached its maximum point, and your protein is at a highest purity level. So now, these are the questions that hopefully we have answered in the course of this presentation, like why does specific activity increase after each step? What does the SDS page tell us about the presence of contaminants? Why use IEX before SCC? If Western blot shows a strong band, but activity is low, what could we do? What went wrong with it? Or what factors might affect your enzyme activity as well? This was the whole point of this presentation. For protein purification, we added a case study as well in it, but mostly it was focused on the techniques that are associated with your protein purification. Thank you.